I want you to take a second and just visualize the blood vessels in your body.
Yeah, if you've ever glanced at a biology textbook, you probably have a very specific image in your head.
Right. You have the red tubes for the arteries and the blue tubes for the veins.
Exactly. And for the longest time, we've thought about those blue veins as essentially just simple biological plumbing.
Like rigid pipes, just gravity drains where, water goes in and water goes out.
It's an incredibly pervasive view, mostly because it makes intuitive sense, right?
Sure, it's easy to visualize.
Yeah. We've taught anatomy for over a century with this neat categorical division. The heart pumps, the arteries flex and push the blood, and the veins, well, they just passively act as the return pipes. Clean, simple, and visually easy to diagram.
But the human brain doesn't really care about our simple diagrams.
Not at all.
When you zoom in on the vascular system protecting your brain, that whole traditional plumbing model just completely falls apart.
It really does.
We aren't dealing with passive pipes. We are looking at a biological landscape inside your head that is, it's actively shape-shifting.
The paradigm shift here cannot be overstated. We are looking at a February 2026 paper published in Nature, and it's titled, Highly Dynamic Dural Sinuses Support Meningeal Immunity.
It's an amazing paper.
It really is. The researchers essentially just threw out the textbook on the neuroimmune interface.
And that is our mission on this deep dive. We are taking that old assumption that the veins around the brain are just passive gravity drains, and we are going to dismantle it completely.
Piece by piece.
Right. I want you to imagine the blood vessels around your brain right now. What if those pipes were actively flexing like muscles?
Just pumping away.
Exactly. What if they could actually reorganize their own cellular structure on the fly to open hidden doors for your immune system? Or lock themselves down to fight off a viral infection.
Because that highly orchestrated chaos is actually happening inside your skull at this very moment.
It's mind-blowing. So to appreciate just how wild this system is, we need to locate exactly where this is happening.
Right. So the protective wrapping around your brain and spinal cord is called the meninges, and it actually has 3 layers.
But we are focusing on the very outermost layer, right?
Yes, the duramater, which is this tough, fibrous membrane glued right against the inside of your skull.
And embedded in that tough layer are the dural sinuses. These are large, valveless veins.
Yeah, and for decades, the medical consensus was simply that these sinuses act as the brain's gutter system.
Like rain gutters on a house.
Exactly. They sit up there, collect the deoxygenated blood and the cerebrospinal fluid, and just let gravity drain the waste down into your jugular.
But the researchers decided to challenge that assumption.
Right.
They used something called intravital microscopy.
Yes. They essentially created a tiny transparent window into the skull of a living mouse.
Wow.
Yeah, it allows them to watch the cellular behavior of these vessels in real time in a living, breathing subject.
And what they saw immediately broke that whole gutter theory.
Completely shattered it. They found that the dural sinuses are wrapped in a contiguous layer of smooth muscle.
Wait, hold on. A layer of muscle on a vein. That kind of crosses the biological streams, doesn't it?
It really does.
Because I thought smooth muscle was strictly the domain of arteries, to help them pulse and push blood from the heart.
You're hitting on the exact reason this is so groundbreaking. Veins, especially valveless ones in the head, aren't supposed to have an extensive network of alpha-smooth muscle actin.
That's the alpha SMA, right?
Exactly. The presence of this muscle means these vessels are not just passively sitting there. They possess the mechanical hardware to actively squeeze, dilate, and pump.
Very much like arteries.
Exactly like arteries.
Okay, let's unpack this. So the passive metal gutter on your house is, it's actually a robotic hose that can squeeze and expand on command.
That's a great way to picture it.
But a muscle doesn't just flex for no reason. Something has to be driving that contraction.
Right. The driving force comes down to a highly specific chemical pathway. The researchers mapped this out and found the muscle movements depend on a receptor called RAMP1.
RAMP1.
And to prove it, they introduced a peptide called CGRP.
Which is pretty famous, actually.
Yeah, it's a well-known molecule often associated with pain signaling. It's actually a major player in migraines.
Oh, wow. So what happened when they added the CGRP?
When they applied CGRP, the dural sinuses actively dilated. They physically widened.
And I imagine they tested the reverse.
Yeah.
Like what happens if you block it?
They did. They used a chemical compound called BIBM 4096.
BIBM 4096. Sounds like a droid from Star Wars.
Yeah, rather than getting bogged down in the alphanumeric soup, just think of this compound as a chemical clamp that blocks the RAMP1 receptor.
Okay, a clamp.
And the moment they applied this blocker, the sinuses vigorously constricted.
They seized up.
Yes, instantly.
So just to kind of recap this first massive shift for you, we've got these dural sinuses, which we used to think were just passive drains. But they are actually active, smooth, muscle-lined vessels controlled by these RAM1 pathways.
Exactly. They are constantly pumping.
Which leads me to ask you, the listener, have you ever thought of the veins in your head as active flexing muscles? Like, how might this change how we view things like head pressure or even tension headaches?
It changes everything about how we understand fluid dynamics in the skull.
Right. Because if I'm piecing this together, it means the veins in our heads are constantly actively flexing, which introduces a massive mechanical problem.
The pressure.
Yes. The skull is a closed, rigid bone box. If you start expanding and squeezing large vessels full of fluid inside a fixed volume, the pressure has to go somewhere.
The thermodynamics of the skull are absolutely ruthless. Intracranial pressure is one of the most tightly regulated forces in the human body.
It has to be.
Yeah, if these vessels are actively pumping, they need a way to manage the sheer volume of fluid displacement so they don't accidentally crush the brain tissue they're supposed to be protecting.
So how do they handle the load? Because it seems like a huge evolutionary hazard to have pumping muscles inside a sealed container without, like, a pressure release valve.
The architecture they uncovered to handle this is breathtaking. In the mouse models, the superior sagittal sinus, which is the main venous channel running right down the center of the top of the head, well, it isn't just a single tube.
It's not.
No, it's bifurcated. It's actually a split-level system with two distinct chambers.
Oh, wow.
There is an upper sinus nestled directly inside the porous bone of the skull and a lower sinus sitting below it in the dura mater.
So it's like a split-level highway.
Exactly.
But for a pressure system to work, the fluid can't just sit there. The chambers have to communicate.
Right. The bone separating them isn't solid. It's riddled with these connecting channels called anastomoses.
Anastomoses.
Yeah. And this is where the fluid dynamics become completely counterintuitive. The blood in the upper chamber flows forward rostrally toward a vascular hub at the front of the head.
Okay, forward on the top level.
But the blood in the lower chamber flows backward caudally toward the back of the head.
Let me stop you there because I'm trying to visualize this.
Right.
We have traffic moving in opposite directions on the top and bottom levels.
Yes.
But earlier we said this is a gravity drain. Does the blood just spill down from the top to the bottom?
Actually, the data points to the exact opposite. Blood is actively moving upward from the lower sinus through the bone channels and into the upper sinus.
Wait, upward.
Yes, it is defying gravity.
That makes no sense under the old passive model.
Yeah.
Water doesn't flow uphill unless there is a pump or, I don't know, an immense pressure gradient forcing it up.
Exactly, which proves these sinuses are dynamically regulating pressure.
Okay, that is brilliant.
The researchers designed an extreme test to demonstrate this. Through that thin skull window we mentioned earlier, they took a microscopic tool and physically compressed just the upper sinus chamber.
Basically temporarily choking off that forward flow. If you pinch off the top release valve, all that blood being forced upward suddenly hits a brick wall. Did the pressure spike?
Instantly. Intracranial pressure skyrocketed to a level that, if maintained, would cause agonal breathing and kill the subject within minutes.
Oh my God.
But the moment they lifted the vertical compression, the pressure stabilized perfectly. This dual chamber system is acting as a vital, life-saving pressure release mechanism.
That is wild. It implies that every second of every day, this smooth muscle network is making microscopic tweaks to the vessel diameter.
Every single second.
Just pushing fluid between these two chambers to keep our brains from being crushed by their own fluid.
Now, humans don't have the exact same bifurcated split chamber structure that mice do.
Right, our skulls are a bit different.
But we have...
So just to give you another quick recap here, the sinus system features these specialized chambers and pathways that act as a crucial pressure release valve for intracranial fluid.
It's an elegant engineering solution.
Truly. So think of a pressure cooker. What happens if the release valve is blocked? That's what your brain is constantly managing.
Exactly.
But, okay, we've established they are mechanical marvels for handling pressure. But these sinuses are sitting right at the absolute edge of the brain, bordering the skull's bone marrow.
Yes, right at the border.
And in biology, borders usually mean security checkpoints. The brain is, the VIP of the body. It is notoriously locked down by the blood-brain barrier to keep toxins and pathogens out.
The blood-brain barrier is famously impenetrable deep inside the brain tissue. But at the outer border, at the dural sinuses, the rules change entirely.
How so?
Well, to see how the scientists performed single-cell RNA sequencing on the cells that line the inner walls of these sinus cavities.
These are the sinus endothelial cells, right? SECs.
Yes.
And RNA sequencing is basically pulling the architectural blueprints for the cell. You get to see exactly what proteins it's being told to build.
Right. And the blueprints for these SECs are entirely unique. They produce high amounts of a protein called VE-cadherin and a specific marker called PV1.
Okay, VE-cadherin and PV1.
But the most important finding was actually what was missing. The SECs completely lack a gene called KLDN5.
I feel like I'm playing biological detective here, because in normal brain vessels, KLDN5 is the gene that manufactures tight junctions, the cellular superglue that seals the blood-brain barrier shut.
Exactly.
So if these SECs are missing the blueprints for the superglue, their walls must be leaking.
Leaking is almost an understatement. Because they lack tight junctions, the walls of the dural sinuses are highly fenestrated.
Fenestrated?
Yeah, they are literally riddled with tiny windows or diaphragmed pores.
So instead of a solid, impenetrable concrete wall protecting the perimeter of the brain, these cells form something closer to a chain-link fence.
A highly porous chain-link fence, and they actually measured the flow rate across this fence by injecting tiny fluorescent microspheres into the bloodstream.
Just to see how fast things could slip through.
Exactly. They watched under the microscope as these spheres zipped out of the sinus, through those tiny windows and into the surrounding tissue, the sinus space.
How fast?
At speeds of roughly 1 micrometer per millisecond.
That is a staggering rate of exchange for a border that is supposedly protecting the brain.
It is very fast.
Which brings up a massive paradox for me. Why would an organ that goes to such extreme lengths to build the concrete blood-brain barrier internally intentionally maintain a leaky, porous chain-link fence on its outer perimeter?
Right.
It feels like leaving the front door of a bank vault wide open.
It seems completely reckless until you look at what is sitting just outside that fence. The sinus space is absolutely packed with white blood cells.
Oh, so the security guards.
Exactly. In a security system, form follows function. The fence isn't porous by accident. It's porous so the security guards can do their rounds and scan the blood for threats.
Okay, that makes sense.
And this leads to what I consider the most visually stunning discovery in the paper. The researchers called it ruffling.
Wait, ruffling? Ruffling sounds way too gentle for a biological defense mechanism. It sounds like a cellular dance.
The dance is actually the perfect analogy. These endothelial cells that make up the fence are not static. They are constantly reorganizing their own internal scaffolding.
How do they do that?
They use a mechanism called the RHO-GT pace cycle. Think of this cycle as a microscopic construction crew inside the cell, continuously tearing down and rebuilding the cell's structural skeleton.
So the fence is constantly unzipping itself, shifting its shape, and zipping itself back together.
Yes, and they are doing it to accommodate specific immune cells. Myelomonocytic cells, to be exact.
Myelomonocytic cells.
Right. When these immune cells patrol the perimeter and need to investigate the bloodstream, the endothelial cells literally shift their scaffolding to create large amorphous pockets.
Ruffles.
Exactly. The immune cells squeeze into these transient spaces, the ruffles, embedding themselves deep into the vein's wall to sample what's passing by.
I want to push back on this a little bit, just playing devil's advocate.
Sure.
Are we absolutely sure this is a cooperative dance? Because immune cells are aggressive. Could it be that they are just brute forcing their way into the wall, tearing holes in the tissue, and we're just calling the resulting damage a ruffle?
The researchers anticipated that exact skepticism. If it's a coordinated dance, there has to be communication.
Right.
The immune cells must be flashing a chemical badge to request entry, and the fence must actively decide to open the gate.
Okay, so did they find the badge?
They did. They injected antibodies to neutralize specific adhesion molecules called LFA1 and VLA4 on the immune cells.
Ah, so they confiscated the badges.
Exactly. And the moment the badges were gone, the ruffling stopped entirely. Within an hour, the immune cells were bouncing off the walls, unable to adhere, and the endothelial cells stopped shifting their scaffolding.
Wow.
But the scientists took it a step further to isolate the mechanism on the fence's side, too. Remember how the smooth muscle pressure was controlled by the RAMP1 receptor?
Yeah, RAMP1 controlled the physical squeezing of the pipe.
Well, the shape-shifting of the fence itself is controlled by a sibling receptor, RAMP2.
Oh, interesting.
When the researchers introduced a highly targeted drug called AM2252, which shuts down the RAMP2 receptor, the results were dramatic.
What happened?
The entire cellular wall froze. The dynamic shifting boundaries became completely static. The gates were welded shut.
Which completely eliminates the brute force theory. The vein itself is an active participant.
Yes.
It is deliberately opening and closing its own physical structure in response to chemical signals from the immune system.
It is a breathtaking level of cellular coordination.
Okay, so just pulling this part together. SECs form a highly porous, fenestrated wall. And they use this process called ruffling, which is mediated by RAMP and those integrins, to constantly open and close gaps for immune cells to patrol.
Perfectly summarized.
So imagine the fence can actively open gates the moment a guard flashes a badge.
Mm-hmm.
How amazing is that cellular coordination happening in your head right now?
It's incredible.
But, you know, it's a beautiful system during peacetime. You have this dynamic pressure valve managing the fluid and this shifting, ruffling fence letting the guards check the perimeter.
Right.
But biological systems aren't tested in peacetime. I really want to know what happens when the alarm bells actually ring.
Which brings us to the ultimate stress test. The researchers decided to see how this shape-shifting border handles a live invasion.
Okay.
They injected the mice with a pathogen called LCMV lymphocytic choriomeningitis virus directly into their circulation.
A virus that specifically targets the meninges. So the threat travels up the bloodstream and hits the superior sagittal sinus. How does our chain link fence react?
The system goes into absolute overdrive. The moment the virus is detected, the baseline ruffling explodes into massive structural shifting.
Like full red alert.
The gates just fly open. The researchers tracked a specific wave of engineered virus-fighting T cells, called P14 T cells, as they rushed to the sinus wall.
Okay.
The entire perimeter actively reshapes itself to pull these specialized T cells and local myelomonocytic cells into the fray. The terrain itself shifts to help the immune cavalry engage the virus.
The border is actively fighting back by changing its shape. But, well, we know how scientists operate.
Oh, yes.
Once they find a beautiful life-saving mechanism, they try to break it to see just how essential it really is.
Yeah, truly.
Did they use that AM2252 drug to freeze the doors shut during the viral attack?
They executed the ultimate sabotage. Right in the middle of the active viral infection, they administered the RAMP2 blocker over several days. They deliberately shut down the brain's dynamic defense system while the virus was storming the gates.
If the fence is frozen and the scaffolding can't shift, what happens to those P14 T cells trying to fight the virus?
The immune response essentially collapses. Because the endothelial boundaries became rigid, the protective T cells lost their mobility.
It got stuck.
They physically slowed down. Instead of being guided into targeted attacks against the infected cells, the T cells were observed just moving randomly along the sinus wall.
Oh, man.
They were slipping and sliding against a closed door, unable to properly embed or engage the enemy.
Because the terrain refused to shift to accommodate them.
Exactly. Without the ruffling, local antiviral immunity was profoundly compromised, the T cells couldn't do their job, and as a direct result, the virus easily bypassed the static defenses and invaded much deeper into the meningeal tissue.
So, synthesizing this for you, during a viral attack, SEC ruffling goes into overdrive to fight the infection.
Yes.
And blocking this RAMP2 dependent process allows the virus to just completely bypass the brain's defenses.
It's a fatal flaw if it's blocked.
Which asks the question, if a virus tries to invade your nervous system, do you want a rigid static wall or a shape-shifting border that actively fights back?
I think the data shows that a static wall is actually a massive vulnerability. At the dural sinuses, the most effective defense is supreme flexibility. The ability to shift, ruffle, and actively manage the terrain is what keeps the virus out.
The implications for how we view neurological vulnerability are just staggering.
They really are. The textbook page showing veins as simple blue drainage pipes needs to be completely rewritten.
Thrown in the trash.
Yeah. The dural sinuses are a highly sophisticated, programmable neuroimmune interface.
We have the RAMP1 pathway driving muscle contractions to perfectly balance intracranial pressure.
And we have the RAMP2 pathway driving this elegant, ruffling dance to orchestrate immune defense.
Which leaves us with a truly provocative thought to walk away with. If this border isn't a brick wall, but rather a dynamic, programmable interface with specific chemical locks and keys, could this be the future of treating brain diseases?
The therapeutic possibilities are enormous. If we can master the RAMP signaling pathways, we might not have to struggle to forcefully push drugs across the blood-brain barrier.
Right. What if, instead of trying to break through the wall, we design a drug that simply tells the RAMP2 receptor to artificially open those ruffling gates?
Exactly.
Could we instruct the brain to actively flush out the toxic protein buildups that cause Alzheimer's disease?
Or consider the reverse scenario. A patient comes into the ER with a severe systemic blood infection sepsis.
Okay, yeah.
Could we administer a compound that temporarily clamps the RAMP receptors shut, freezing the dural sinuses and hermetically sealing the brain off from the infected blood until antibiotics can clear the system?
We are talking about having manual override control over the brain's security system. It is absolutely mind-blowing.
It's the frontier of neurology.
Thank you so much for taking this deep dive with us today. I can guarantee you will never look at a diagram of the human body the same way again.
Definitely not.
The next time you feel a pulse in your head, or picture the blood flowing around your brain, remember, you don't just have passive plumbing up there. You have a flexing, shape-shifting, heavily armed security perimeter working tirelessly to keep you safe.